All right, let's dive into something that sounds like it's straight out of a sci-fi movie, but I promise you it's very, very real. We're talking about gene editing. Yeah, the ability to literally rewrite the code of life. It's not a what if anymore. It's happening right now. The big question is, how on earth is that even possible?
Okay, so to really wrap our heads around this, let's just push all the complicated science stuff to the side for a minute. Instead, I want you to think about something you probably do every single day: typing on a computer.
I mean, just imagine it for a second. What if you could edit DNA, the very blueprint of life, just like you edit a document? You could just scroll through the billions of letters of genetic code, find one tiny little mistake, a single typo that might be causing a devastating disease, and then just backspace, delete, and type in the right letter. What if?
Well, that's not a what if anymore. That is exactly the incredible promise of the tool we're going to break down today. It's basically a biological find and replace for our genes, and it's called CRISPR.
So a tool this powerful, this precise, you'd probably think it was engineered from the ground up in some super high-tech lab, right? Well, the story is actually way cooler than that. The secret to this revolutionary tech, it comes from a tiny, ancient war that's been raging for billions of years: the battle between bacteria and viruses. Okay, let's just get this out of the way. The name, CRISPR, is a total mouthful. It stands for Clustered Regularly Interspaced Short Palindromic Repeats. Yeah, I know. But don't get bogged down in that. All you really need to know is that these are these weird, repeating patterns of DNA that scientists first spotted inside bacteria. And for the longest time, nobody had a clue what they were for. And this whole thing didn't just pop up overnight. The story really starts over 20 years ago with a biologist named Francisco Mojica. He was studying some pretty unique bacteria from a salt marsh in Spain when he kept seeing these bizarre, repeating bits of code in their DNA. He was the one who was like, wait a minute, what is this? Eventually, Mojica cracked the case. It turns out CRISPR is a kind of ancient, brilliant immune system for bacteria. Here's how it works. When a virus attacks a bacterium, if the bacterium survives, it actually grabs a little piece of the virus's DNA and stores it in its own genetic code. It's like taking a mugshot of the enemy. So if that same virus ever tries to invade again, the bacterium has this most wanted poster on file. It can recognize the intruder instantly and just obliterate it.
And that is the light bulb moment. That's when scientists looked at this incredible search and destroy system and thought, hang on, what if we could hijack that? By pulling out the key components, they turned this natural defense mechanism into one of the most powerful gene editing tools humanity has ever seen. So how does this toolkit actually work? Well, it really comes down to two main parts. First, you've got something called a guide RNA. This is the brains of the operation. Think of it like a super precise GPS. You can program it to hunt down and lock onto one exact specific spot along a massive strand of DNA. Then you've got its partner, the Cas9 protein. This is the muscle. Cas9 is basically a pair of molecular scissors that just follows the guide RNA around, ready to snip the DNA exactly where it's told to.
Okay, so we've got our search function, the guide RNA, and our cutting tool, the Cas9 protein. So how do we put them together to actually edit a gene? Well, you might be surprised at how elegant and honestly, how simple the process is.
What you're seeing here is a fantastic look at the whole system in action. You can see the complete CRISPR-Cas9 complex zeroing in on a piece of DNA. It looks complex, but the idea is simple. You're seeing the toolkit, finding its target, getting ready to make the cut, and then preparing to replace a piece of the code. Let's break it down.
It all happens in 3 simple steps. First up is find. That guide RNA we talked about, the GPS, it zips along the DNA strand until, bam, it finds the exact sequence it was programmed to look for and latches on. Step 2 is cut. The Cas9 protein immediately gets to work and makes a clean slice right through the DNA. And finally, the coolest part, replace. The cell has its own amazing natural repair kit. It sees the broken DNA and rushes to fix it. And right at that moment, scientists can slip in a new corrected piece of DNA for the cell to use. And just like that, the code is rewritten.
And that's it. The edit is done. The original, maybe faulty gene is now fixed or replaced with a new one. It's literally a find and replace function, but for the very source code of a living thing. Pretty mind-blowing, right? So, this all leads us to the most important question of all. Now that we have this incredible power, what do we do with it? The potential here is just staggering. We're talking about applications that could completely change everything we thought we knew about medicine, about food, about life itself.
I mean, the possibilities are almost endless. For researchers, it's a dream come true. You want to know what a gene does? Just use CRISPR to switch it off and see what happens. But it gets more profound. We can now go in and correct the genetic typos that cause horrible diseases. And this isn't theoretical. The very first CRISPR treatments for diseases like sickle cell anemia are being used on patients right now, essentially offering a cure. We can disable genes that viruses use to infect cells, or we can re-engineer crops to survive drought. It's a total game changer.
So, there you have it. A defense tool borrowed from the humblest of bacteria has given us an almost unbelievable level of control over genetics. It's an amazing testament to why we need to keep exploring basic science. You never know where the next revolution will come from. But it also leaves us on the edge of a new era, with a massive question hanging in the air. Now that we have the power to rewrite the code of life, where do we go from here?